to, again, submit sequences as well as to do queries and retrieve sequences. So entries for NCBI, get entry for the Japanese database, and EBI search for EBI. And I guess it's just to emphasize that aside from retrieving sequences, you can also submit sequences to these portals. So I mentioned before that databases can be classified into two, primary and secondary databases. And the primary databases, the content is controlled by the submitter. So whatever content or information the submitter submitted is what is found in the primary database. Now, from the primary database, that can be further used for assembly, for example, or for curation, according to, again, to the different functions of the genes, or to remove possible errors or to assign functions to what was submitted here. And so you come up with your secondary database. And most of the work is done by bioinformaticians that are employed by the NCBI. So GenBank is NCBI's primary sequence database. So it's a nucleotide-only sequence database. That means there are links to protein sequences, but it is nucleotide that are deposited in GenBank. So we're going to see the different types of nucleotides. Again, it is mainly for archival in nature. And again, you can see the different revisions of the sequences submitted at different dates. And one deficiency of GenBank is the deficiency of primary sequence data of primary databases is there's a lot of redundancy. Again, different labs working on the same bacteria, depositing the sequence of the same gene, okay? And they're all gonna have the different names of submitters. So the records in GenBank are divided into 18 divisions. There are 12 traditional and 6 bulk records. And for the traditional, you can see the taxonomic division of those different records. So you have primate, plant and fungal, bacterial, viral, even a phage. And there's also sequences from your cloning vectors, from your plasmids, from your metagenomes, from environmental samples, right? And the traditional divisions tend to be accurate, and it's accurate compared at least to the bulk sequences. They're more characterized because most of the information that's deposited in them, there's a few genes, usually one or a few genes. And so it's easy enough for scientists to check for accuracy before they submit to these databases. The bulk, on the other hand, from the name, it's bulk. That means it's a lot of sequences. And because it's a lot of sequences, there's just too many sequences to look at individually, okay, to check for accuracy. And examples of this are your express sequence tags. We're gonna see more detail of that, what that is later on. So, so these are like massive sequencing efforts. You sequence a lot of DNA fragments or cDNA, a lot of cDNA, a lot of clones. And patent when they also sequence whole organisms. And the sequences from all those genes are deposited as a patent. Although we know now we know now, we have already learned from regulatory that we cannot patent sequences because they occur in nature. Okay, it used to be, yes. Yeah, I'm not sure if I've mentioned, I used to be a co-inventor in several patents, but they are patents on fungal sequences. But with the new ruling that you cannot patent sequences, pretty much my co-inventorship is gone in all those. Okay, we're gonna go through this later on, so. But pretty much the guiding principle, the records are grouped for various reasons, and the reason for that is really to be able for users to take full advantage of databases in terms of, again, finding information. So the GenBank, again, is our primary NCBI nucleotide sequence database. And as I want to point out here, I think it just, it does describe the type of database it is, the format. So it's, the records are in a flat file format instead of relational tables. And that's right. And we're gonna explore GenBank. Come explore GenBank with me on this rainy day. Okay, where do you find all these different divisions in GenBank? So genomes, it's actually these headings here that we're gonna go over first. So yeah, submission tools and how to submit things. As I mentioned, Bankit is now the major one that is being used to submit sequence information. Sequin, they're getting rid of that. But here is where genomes is. So these are for complete genomes from different organisms. That means all the sequences have been edited. Okay. The next one is and assembled into a full genome, by the way. That's the important part is it's been assembled into a full genome. But again, this is GenBank, so there's redundancy. So for a different for one microbe, if there are several labs that sequence one species of bacteria separately, then all that information are to be found in this database right there. Then there's also the whole genome shotgun, and it does say that, so I guess from here again, it's contigs from ongoing whole genome sequencing projects. So it's not completed genome sequencing projects, they're still ongoing. What are contigs? Contigs is short for contiguous. So it's contiguous DNA sequences. So yeah, that's the thing with this feed here, you cannot use the board because they won't be able to see the board. But for example, so I'll do my best to, let's say this is, so you clone a genomic library in your cloning plasmid, okay? And you sequence all of them. That's, by the way, shotgun fragments is they isolate chromosomal DNA and then they just use physical methods to chop the DNA into a certain range of fragment size, like 500 to 800 nucleotides long. And then those 800 nucleotide fragments, they sequence all those, okay, to come up. And so let's say this is one 800 nucleotide fragment, and then they found another one. And here's another one. And again, by looking at the sequences, using their bioinformatics tools, they found that there's an overlap with this other fragment. And they found another fragment which, again, has an overlap with this fragment. Has an overlap with this fragment. Let's just say this is a longer fragment that they sequence. And then they found another one which overlaps with that fragment. And they have a lot of this, maybe 50, 100, 200 fragments that, again, using their bioinformatic tools, they were able to find sequences that are identical. So they were able to overlap that. And so a contig is essentially the assembly of that overlapping sequences. So the contiguous sequence. So this would be a contig that is based on the sequences of these 4 overlapping sequences. And again, that can be 50 or 100 or more than 100 overlapping sequences that are used to assemble a contig. And again, you just get an idea here as to the location of different genes with respect to each other. And again, when you're mapping genes that may be responsible for a certain phenotype, maybe in humans, in the chromosome of humans, if you don't have the complete genome sequence yet, then having these contig maps would help in trying to find the physical locations of those genes. And to get to this whole genome shotgun projects to find out what's going on now. So you just go here. And this gives a complete list of all the whole genome sequencing projects that are going on. And you can see here the create date and update date and the total length in megabases. And there's also a column on whether it's got annotation or not. Yeah, so this Stenotrophomonas is the bacterium that is used probably in BTEC 3100 or 4300, or you will encounter in your capstone project. And so you can use, if you want to find out what genome sequencing projects are going on and which labs are doing genome sequencing of Stenotrophomonas. Yeah, this is the bacterium that is involved in degradation of organophosphate pesticides. And you can see here, and you can see that there are many Stenotrophomonas, let's say multiphilia, and there are many different strains of that. And it's possible that it may turn out that there's almost no difference in the genomes of those different strains. Different labs may be working on it, but they may actually be working on the same strain. They're only going to find out once they have finished the whole genome. So those are not yet completed genome. And once the genomes are completed, then it goes to the genome tab. Okay, high throughput genome. This is even more unfinished than the whole genome sequences. So these are even smaller fragments that have not been assembled that much into context. So there's still a lot of gaps in the sequences. So again, if you're looking at here, you're maybe looking at individual ones of these without any gaps. They're just everywhere. And yeah, in terms of being able to use this, this is one that you cannot find much use for unless you get a specific need for it. And there are two types of high throughput genome sequences in GenBank, unfinished record. And this will remain unfinished. They pretty much stop doing any sequencing. And the finished record, of course, they have plans to finish it. So once they are finished, and if they're unique, they might end up in the genome's database here. Express sequence tags from the name expressed. When we say express in terms of nucleotide, what type of nucleotides are we looking at? mRNA, right? mRNA or RNA, okay? So remember first that now there's technology that can sequence an RNA strand directly, okay? But it used to be you cannot sequence RNA. Okay? You can only sequence DNA. So the information from mRNA before, we obtained that by reverse transcribing messenger RNA. Okay, again, recall reverse transcription from your other courses, from your 1322 and other courses. So essentially, here's your messenger RNA. You use a primer, and then you add reverse transcriptase. And what reverse transcriptase does is it uses the DNA or the mRNA. Okay, let me say that again. It uses the mRNA as a template for the synthesis of a complementary strand of DNA. Okay? It's reverse transcriptase because in our non-reverse transcription, we use the DNA as a template for RNA synthesis. Here, RNA is used as a template for DNA synthesis. Okay? And then you get rid of the RNA by using RNase, ribonuclease, and then you synthesize the second strand now of your cDNA, and so now you've got a complete cDNA, and this is now what they subject to our Sanger sequencing or other next generation sequencing. So what are express sequence tags then? So they are single reads from mRNA. So you have a bunch of this cDNA representing different mRNA from different genes. And so they just sequence the ends of this cDNA. And so those are the express sequence tags. And again, since like we said that we have 3 billion nucleotides in our human genome and only around 100 million of it actually codes for proteins. That means they are transcribed. You're not able to assemble a whole genome from sequencing cDNA or express sequence tags, okay? Because the introns, for example, won't be found in the sequences of this. And besides, again, if you have a cDNA which is, let's say, 2,000 kilobases, the express sequence tag would only be able to sequence, let's say, the end of that, one end, and another one here. So those are about these two are about the only sequences that will be found in your express sequence tags. There's a lot of sequence that won't be found in your EST database. And again, when we look at our GenBank flat file, we did go over the type of information, and in the locus, it does say there what kind of biomolecule it is. So in this example here, it does say it is an EST. And one other characteristic of ESTs, it's poorly characterized. Remember, in an example of GenBank file we looked at before, the start codon is described, the stop codon, and also there's possible promoter regions, terminator sequences. And again, because this is really incomplete sequences, there's not much in terms of our feature, much of the features that are included here. And the transcriptome shotgun assembly sequence database is similar to the contigs, but it's the contigs for the genomic DNA, but this time it's overlapping sequences from sequencing of cDNAs. And there's something similar in terms of equivalent to ESTs, but instead of cDNA, it's actually genomic DNA that are sequenced, and it's the ends of the genomic DNA fragments that are sequenced. So now moving on to secondary or derivative databases, and again, this came out from further working on the sequences that are found in the primary databases. So here, the GenBank updated only by submitters, and then the third-party bioinformatician from NCBI who will curate it, maybe remove redundant sequences to come up with the RefSeq, or perform some more genome assembly. So essentially, they're cleaning up what is found in this messy primary database, okay, and also giving them ascribing functions to certain of the sequences, and maybe grouping those different genes according to biochemical pathways, shared biochemical pathways. And so here's our nTrace protein database. And so to go to our nTrace protein database, we want to do searches there. And again, we're not gonna be submitting sequences, we're gonna be just doing searches. So here again, the different databases in NCBI. And so this is where we're gonna find protein. And so in today's exercise, we're gonna be searching for different proteins given the names. And so for example, we're gonna search for DNA mismatch repair, okay? So again, just do this on your own so that you're gonna get practice. Practice makes perfect. DNA mismatch repair. And we got a lot of sequences, and you can see I didn't specify. Here it says Homo sapiens, and I didn't really specify the organism. How can I limit my search then to humans? Anyone recall PubMed? How are we able to limit the fields which are searched
filters.
How do we go to those fields or filters? Advanced, right? Okay. So yeah, here you can specify. So humans, what field will humans be in? Organism, right? Okay. So and again, we now also have RefSeq. Well, let's just say, okay, so this is my search of DNA mismatch repair. Okay, let's limit it to fields to humans. So first we add that to our search builder. Come on, man. And then we said what field for humans? Organism. Yes, there's an organism field right there. And so we can type Homo sapiens or just see if humans is in the index list. Human, right there. Then we search. And so these are the records which shows DNA mismatch repair and human. And then RefSeq, reference sequence. Again, reference sequence are the non-redundant records. Okay. So of these results, how many are non-redundant results? Where do you see RefSeq? You find it here in the source database. Okay. So familiarize yourself again, if you're in protein, what are the different filters that you will find on this left side here? Okay, so RefSeq is right there. And when we say how many of them are non-redundant results, then you have 182, which are non-redundant. Okay. And how do you tell the non-redundant records? You can tell that they are non-redundant records because they start with an N for non-redundant, right? So here it also shows the accession number for non-redundant mRNA. So NM, M for mRNA, P for protein, genomic sequence, okay, chromosomal sequence, contigs, and WGS super contigs. We're gonna be working a lot with NG, NM, and NP. Okay. And it also talks about predicted. So this would have been probably shown experimentally that they code for proteins, okay, while these are computational prediction of the presence of proteins. So again, they all start with N, or they might have an XX that you would see there, XP, because this is protein. But if we unclick the RefSeq again, and look at the accession numbers, So if they're non-reference sequences, then they don't start with an N. And the benefits, I already mentioned, RefSeq benefits, non-redundancy, and it links nucleotide and protein sequences. We're gonna see how we're able to link nucleotide and protein sequences through N-trace. And now for the other protein sequences, protein databases, we have UniProt, the Universal Protein Database, and it integrates pretty much the records found in SwissProt and Tremble. And so if we search for UniProt, and you can see here that the records found in SwissProt and in Tremble are actually also searchable in UniProt. So Swiss prot are like manually annotated, and while Tremble, this is computationally translated. So we also have GenPep in, that's right. We also have many specialized protein databases. So antimicrobial peptide sequences from different organisms, plant, animals, bacteria. This is G protein coupled receptors, a certain membrane protein that's involved in many, implicated in many disease, and also as targets for drugs, like for addiction. Immunogenetics, it's like sequences of antibodies, and so on and so forth. And we already have Uniprot. And we're gonna use this in much more detail later on. So this is just to introduce to you the different databases. So SwissProt again is manually annotated. It's not redundant, but it's manually annotated. So there's fewer entries. So look at the number of entries in SwissProt, 557,000. Now this is computationally translated. So a computer can just crunch out in those translations 120 million. So and again, SwissProt plus Tremble equals all known protein sequences. For the protein structure databases, we want to be able to model the tertiary, quaternary structure of proteins to be able to develop drugs, or to be able to find out how certain, let's say, estrogen binds to an estrogen receptor, and probably how to design cancer drugs that will interfere with estrogen binding to the estrogen receptor, and so on. So we have in NCBI, we also have a structure database. And again, this is how you go to the NCBI. I guess you can tell from here. Structure database. And this brings you to, again. And there's other, like programs that are used to determine the structure of different proteins. So molecular modeling database also, C and 3D. And you're gonna be able to see this in today's exercise. Actually, that's the structure using C and 3D. Later in the course, we're gonna use a program to actually also determine which amino acid residues interact with, let's say, again, go back to estrogen, the estrogen molecule. And we also have the protein data bank, which would also show the 3D protein structures. Conserved domain database. So for a particular protein, for example, one protein that is a transcriptional regulator, there's a domain that is involved in DNA binding, and there's a domain that is involved in interacting with RNA polymerase, okay? Or there's a domain that is involved in interacting with an inducer molecule or a co-repressor molecule. So there are different domains, and you can find those different, identify those different domains to this conserved domain database. Homology, like the homolog, when we say homology, that means it's possible that the two genes, several genes, are derived from the same ancestral gene. Okay. Again, some of our genes are highly conserved down to bacteria. Okay. And so this is the database to identify those genes that are evolutionarily related. And again, of course, there's a gene. Again, this is all found here. And we're gonna use those gene searches later on, the interface. Genome, we're not gonna use that today. But again, oh, here you go. This does show the number of assemblies that are available for these different genome sequencing projects. And there's specialized genomic database. I've already mentioned that. There's, if you're just looking for some mouse sequences, then there's a mouse database and Drosophila, a fly database. And there's also databases based on sequence variants. Again, many of our diseases are just due to one nucleotide change from the normal. And so that single nucleotide polymorphism that, and some of them, so we have many SNPs, okay? And some of them are associated with disease. Some are, we haven't found any association with disease. So all those SNPs will be found in this database. Now, if it's only human, then it's these database variants. Okay, so the many different, for each gene, there can be different SNPs. There can be a change in nucleotide number 15 or a change in nucleotide number 30 in different people. Okay, so those are the database for those SNPs. Now, if they have been, a SNP has been shown to be medically relevant, then you will find that information in this particular database. And the exercise should be ready now. That's right. Actually, I will help you go through this exercise. And yeah, you will also participate. Okay, this one I didn't cover in today's lecture. But you know where to find, how to find databases, right? The NCBI databases.
Yes. Last pictures for this section.
Okay. Okay, scroll down through this database here, and how do we find the scientific name of this particular insect? So scroll down and you'll find taxonomy right there. Okay, and yeah, you're just gonna do this on your exercise, and then you're gonna submit your partial exercise later on. And again, you can write notes on your exercise as to how you got, how essentially the steps that you took to find the answers. Okay, so here is, so what is the scientific name of the Asian citrus psyllid? What's the scientific name? Anyone? This is the scientific name, right? The genus and the species. Okay. Now be careful in your exam. I might ask for the scientific name of the citrus, not the insect. So read the question carefully. Okay. Okay, what is the genus and species? So again, for the exercise, you're supposed to snip your search strategy and your answer to the question. So you use taxonomy. we search for that query terms. And then this is our scientific name. Genus, species, format. Okay. Now, what is its lineage? How do we find the lineage of the taxonomic lineage? Well, there's not much we can click on here. There's nucleotide, there's protein. This one is clickable, so let's click on that. And here it is. This is where we find the lineage, right? So write a note to yourself, click on the scientific name to find the lineage. Okay, the lineage, we already answered that one, taxonomy. We should highlight that one because that's an answer to the question. But this is the lineage. So again, make sure you at least submit a partial submission before you leave class. Okay, now how many nucleotide and protein sequences are available at NCBI for Sept? Okay, in this particular page here, do you find anything that might answer this question? How many nucleotide and protein sequences are available at NCBI? Once you find it, say yes. Yes? Yes? Okay. Where do you find it? To the right, to the left? On the right, the table on the right, right? Correct? So we snip that. Okay, and it says nucleotide and protein sequences. So these are the number of nucleotide sequences and the number of protein sequences. Okay, next question. How many literature on Asian citrus psyllid have full contents? Oh, now, what do we do with this now? Is there anything here which will indicate to us how many literature have free?
Is it PubMed Central?
PubMed Central, right? Again, PubMed Central is that sub-database of PubMed, which the whole article is available for free. And I'm just gonna be lazy and, no, I cannot do that, huh? Okay. You can erase, and then highlight PubMed Central. Or you can circle it if you want. Okay. So based on the cursory examination of the first 20 proteins, that's NCBI protein contain only non-redundant sequences, okay? So we go back here, and this is where we're gonna be able to access the protein records, right? So just click on that. So this is how you're gonna be able to access the nucleotide records, etc. This is where you're gonna be able to access the free literature, okay? And so, again, just play with all the different things on this website. So NCBI protein, and take note, it takes us to protein database. And here's our search term now, okay? And I think I still have this filter here from the earlier search. I'm gonna clear that first. And then it says based on a cursory examination of the first 20 proteins, does a CBR protein contain only non-redundant sequences? Yes or no? Only redundant, or only non-redundant, or is there redundancy?
There's redundancy.
How can you tell that there's redundancy?
It starts with an A.
Yeah, the accession number, right? AX, it doesn't start with NP. We said that if it's non-redundant, it's N as in non-redundant, then protein, and it starts with A here. So that's our answer, right? Accession numbers. Many of the accession numbers don't start with an N. We even have NP in the first 20. No, we don't. Okay, how many of the protein sequences from the insect have ATP in the title? Okay, how do we specify, how do we limit a search to those with ATP in the title? Literature with ATP in the title, or sequences with ATP in the title. Where do we go again? If we want to add more filters, go to advanced again, right? And it's title one of the fields. Let's scroll down. Title. ATP. What else do we need to do while we're here?
We can see.
Sorry?
So we can see.
ATP.
Sequence like.
Now you're gonna see that later on. But first, before we do the search, what are we gonna do? Add this, right? And then search. And then where do we now sequence length in the range of the 100 to 1,000 amino acids? This is actually where you do it, sequence length. So again, just familiarize yourself with the filters. So custom range, what's our range? 100 to 1,000. Then apply. Now what happened? To 1,000. Custom range. Title. Did anybody get something different in the number?
Search now.
It's still the same insect, diaphorina. Yes?
What is the first part of your search say? I can't see it.
I'm sorry?
Yeah. It says ATP.
Oh.
He went to like advanced. He typed in ATP.
ATP is the title, right?
Is
that what that is?
So ATP is the title and
Yeah.
the sequence.
Well, no, the first part.
is the range.
Right, and then the second part is just from the
Sorry.
previous search. And so you just
Sorry.
What was that? 'Cause I didn't get that.
it was the taxonomy search. It's
Research.
Yeah, it was when we were trying to find the taxonomy name for like the organism. That makes sense.
Yeah.
Okay, let's try it again.
I would just research whatever is under taxonomy. Find out.
Should have given us records, right? Oh, now it's error. That there's nothing that's being pulled out. Let's look at the sizes of these proteins here. This one is 224 amino acid, 224, 224. Those alone should have been pulled out. So I do not know what's going on, because I made sure that I double check all this. We're gonna try it again later on, or maybe you can try it at home. See if you get a different result.
Too slow.
Are we going to get it wrong?
Huh?
Are we gonna get it wrong?
Yes.
Now, later on, after class, you can try it on your own. Maybe it doesn't want to work right now.
We won't know how to do it.
We got our own.
If I try it this afternoon, it's still the same, then we're all gonna have the same. Yes,
When is this going to be due? Tomorrow.
it does say here that it's gonna be due Tuesday midnight.
We usually have like.
Yeah, why do we have it on Wednesday?
Okay, next one is searching for this gene in humans. Okay, which database are we gonna be using?
I don't know.
Looking for a specific gene. Genome is all the DNA sequences, right? We're just looking for one gene, right? So here. Gene, wait, clear all. Being slow. Gene. Then breakpoint cluster region. Okay. How many records are there on breakpoint cluster gene in humans? Oh, is there something else we need to do? Because I just searched for the gene. Is there something else I need to a filter I need to apply? In humans, right? So you can go to that Homo sapiens, there's 42 there, or do advance. And then organism again. Okay. There's a difference if you click on the filter that's on here and if you do the advanced. Click on the filter here, it just said 42. Here it says 44.
Oh, it has 44.
Did it have 44 earlier? Okay. Okay. What's next? Backwards there, you okay, go ahead. Make sure you go back to your answers and make sure that you answered all the questions that are asked there. I might have skipped some of it. So, okay, what is the accession number of the reference genomic sequence for the gene? So how do you find that? Okay, you click on this. You click on the gene. This is the one which is a BCR. This is actually a fusion protein here, which leads to chronic lymphocytic leukemia. So this would have the full gene. And so we click on that. And what is the accession number of the reference genomic sequence of the gene? So where do you find that information? Again, you study this. Study all the table of contents, the related information that we went over in class, especially. And this is our NCBI reference sequence here, right? Because it's looking for the RefSeq reference sequence. So we click on that and the reference genomic sequence or gene sequence, here it is, right? Reference, it's non-redundant. It starts with an N, G, gene, right? So this is our reference gene. So again, once you go home, practice and be sure that you're able to come up with these answers again. I'm supposed to highlight that. Okay. Now, the accession number of the mRNA transcripts and the corresponding proteins for the gene. Okay. And again, if you just explore this page here, you can see that there's this area here with the non-redundant or reference mRNA sequence, NM, and the corresponding non-redundant protein sequence. Okay. So actually there are two, by the way. There's one here and there's another one here. Okay, and you can see the difference between the two. One is isoform 1 and one is isoform 2. That means there's probably alternative processing of this, okay? Because this is from the same gene and yet there are two different isoforms. That means there's alternative splicing of the RNA and different sizes of the proteins. So mRNA and protein, so this is one. And we're gonna snip the other one too because there's two of them. Yes.
So if we're doing this on the exam and we see like 5 that we have to snip all 5 and post them?
No, I'll probably tell you to just pick the top one. Okay, how many exons and introns are there in the gene? Now this one is not easy to find. You're gonna have to go up there. Okay, and then here pull down the full report here. And go to the gene table. So in your exercise, if you want to get a snip of that so that you will know how to find it, then go ahead and include the snip of that. Okay, now click on the gene table, as I said. And here's the gene table here. So complete gene table. And you're asked how many exons and introns. And if you click on this, this is the exon table for the top one. We're just gonna do the top one. Okay. And looking at this, how many exons are there? Do you see it?
23.
Right there, it says 23 coding exons, right? How many introns are there?
2.
How do you find out how many introns are there? So here is the column of the different exons, different exon lengths. These are the different introns. This is on the page 1. Go to page 2 or the last page. And the exons we said are 23. There's one less intron, so how many introns are there? Louder?
200.
So we said there are 23 exons. And so this is the 23rd exon right there. Items 21 of 23. So how many introns are there?
22.
22, right? So you're gonna have to answer, type there somewhere, 23 exons and 23 minus 1 introns. Okay.
I know how to do that. So you just clicked on the genomic table for
Under here, gene table, right?
gene.
Used to show summary before. Yeah, the summary here.
Thank you.
Okay, are there any human diseases attributed to defects in this gene? Let's go again to our summary. Yeah. Full report. Summary will work. Okay, and in here, are there any human diseases attributed? So if you go back to our full report, where we found our reference genes and reference RNA and protein. And again, familiarize ourselves with what's on here. And it says there, anybody remember the, oh, okay. What are the entry numbers for this in the online Mendelian inheritance in man? So the OMIM is where you will find mutations that have been shown to cause disease in man. Okay, online Mendelian inheritance in man. And of course, they are heritable. And so, are there any diseases attributed to defects in BCR? Okay, so this is where you will find it. Again, I'm back to the gene full report right there. And scroll down and you'll find there's OMIM right there. There's a link to OMIM entry. And there are 3. Okay. So what are the entry numbers for this in the Mendelian database? And so we can just snip that. And what the OMIM numbers are right there. Are you ready for the next one? Number 3. Okay, so protein database again. So we go to our protein database. And search for this protein, for this enzyme. Okay, is there anything else we need to specify in our search to limit our search? Again? Advanced, right? No, we're still in that OMIM thing. Again, make sure you're in the database homepage before you do your. Okay, what filters do we apply? Organism. And okay, are we okay? Don't forget to add. Oh, it's already there. So I didn't click on organism. I like to not complete it to find out whether what I'm typing as a query term is actually found in the index list. And search. And so what else are you asked to do? Filter. Only the ones in the RepSeq protein database, right? So that means you have to click on this. Okay. And there are many different isoforms, and you can look at the isoforms, and you can see that there are different sizes for those different isoforms. Okay, but we're gonna go with the rest are XP, And so we're gonna go with the NP. Okay, so the XP are predicted. The NP is our main ref sequence for protein. So what's the reference sequence? This one, right? What is the accession number for the same protein in UniProt database? Okay, so we're gonna go outside of Entrace now. Accession number, NP. Okay, how about in the UniProt database? So again, you just look as to if you have anything here. And if not, that will take you to UniProt. This one says 0, so maybe that wouldn't work. But this is the protein we're interested in. So let's click on that. And let's see if there's any related information on here. Does anybody see UniProt here? There it is, protein UniProt. Okay. Remove this. Okay, just in case you didn't see what I did, I unchecked this refseq here because it's a filter. And here's the equivalent protein in UniProt. So this is the accession number in UniProt. Next, are there any known 3D structures? From this page, you can already see that there's 3D structures for this one right there, right? On the right side is the protein 3D structures. And so yes, how many are there? It does say there are 3 different structures. So you can click on that and it does show you the 3 different structures. Yes?
Oh, I think I see it. Okay, I got it. Do you want to start turning this in? 120.
Yes, before you leave. Okay, it does say here under gene features. Where do you find the gene features? In a peptide record. Remember, if you scroll down here, is where you will find on the left. It's where you will find, not on the right, on the left. It's where you will find the features. Okay. And what is the name of the gene that encodes for aldehyde oxidase protein? It does say there, the gene, and that's the name of the protein, that's the name of the gene. The designation for the gene is AOX1. Okay, now number 4. Yeah, this should be easy. The authors, how do we find authors? I'll just do it the lazy way, or the fast way, not the lazy. I won't say the lazy way is the fast way. Maturis and Singh and Dreyfus. Let's see how many results we get. Oh, wait a second. I'm supposed to be searching for what? Am I supposed to be searching in the protein database? It just says our 1994 article. So what search engine do we use? What database do we use? PubMed. Okay, so when we search in PubMed, there's only one article, and you can look for the answer for A there. What are the two main ones? And what is the rep-seq accession number for the protein, which is mentioned, the from should not be here, which is mentioned in the title in 1A, okay? So again, this is the name of the protein, right? So you just go to protein and search for the name of that protein, using that name of that protein. So if they do it the other way, it also pulls out the protein from many different organisms. So if you just want the human protein that is described on here, you just go to this link here. And the rep-seq is already chosen. Is there another filter that I need to apply? In the 1994 article, okay, this also pulled out other isoforms aside from the F1. Filter that.
Pastor, it's 126.
Yeah, you can leave now. Bye.
We also have those. Today, yeah. Oh, yours is today? Later.
Which one is yours?
Does anybody know how to see the comments on the Turnitin?
No, it doesn't say anything.
Wait,
Nobody
no
is
one
able-
can see because the guys in the other class told me they could. So maybe you should check again. And,
I just checked the
Has anybody here seen the comments? Raise your hand. Nobody? Nobody's able to see.
I couldn't see mine.
I will check.
He finally gave up the No, you're fine. Okay, then yeah.
They just give me a call or send an email.
Email. Did you hear? People would die.
Yes.
One day.